Hello, this is Nick Cap, and this is my 10-minute review of stem cells for dummies. We want to cover pages 1 through 100, and I'll just kind of go over those pages with you real quick. Here's the start of the stem cells for dummies. There's the author information. As you can see, we're kind of going through this, and we're going to brush up on our biology of stem cells. We're going to talk a little bit about what stem cell science is. I'll lecture on this a little bit, and so that's basically what we want to do. I want you to know the biology, and I want you to know what kind of stem cells are, and that's the reason why we're kind of going through these intervals. Here's a little bit longer table of contents that will go, and so I will just kind of go real quick. And so basically he's just telling you why he is interested in stem cells. This talks to you about how we're going to use the book. There's a number of highlights and stuff. What you're not to read, I'll go through this in foolish assumptions. I'll kind of go through all of this of kind of what to do. Again, usually the chapters will start with a little humorous anecdote and says, "Believe me, if there was an easy stem cell fix for baldness, our team would have found it by now." And again, all these guys are bald here, and it shows that. Again, stem cells, cell research has a relatively long history, of a couple of decades. We really haven't been able to grow stem cells for kind of a really long time. And so what this is looking at is in this chapter number 1, we're looking at the foundations of stem cell science. What biology do you need to know? And so some of you haven't had a biology class, and I think it's real important that you do that. Understanding what researchers need to know, and to look at what scientists kind of still need to discover, and kind of all that's kind of out there. Again, we're going to be working with animals and other organisms, and basically what we need to do is we want to be nice to these organisms. We want to be respectful to these organisms as well. Again, mice are very critical for stem cells. We're going to be using the mice in there. We do everything that we can to kind of prevent pain in the mice or anything else like that. And so again, what we want to do is be real careful with that. Again, this talks a little bit about how cells make skin. There's a bunch of stem cells that are involved in that. We'll look at the immune cells in there as well. And so the immune cells are responsible for determining whether you accept or reject a graft or something like that. Some key properties for stem cells, and we'll kind of go over that. Again, one of the key properties of stem cells is that an embryonic stem cell will become any cell in the body, and that they divide, and it's not of uncontrolled divide. They divide, but they can divide forever. They are basically immortal. We will also look at kind of adult stem cells. For the most part, adult stem cells aren't as totipotent as the stem cells that we see from embryos, but again, we have some way of kind of recreating those cells, what we call induced pluripotent stem cells. Again, how do we confirm that cells really are stem cells? We grow them for several months and ensure that they're self-renewing. We examine the cell surface looking for markers that only occur on undifferentiated cells. We look for the presence of OCT4 protein and other signaling molecules that typically are produced by undifferentiated cells. We inject them into immune-compromised mice, and we see whether they can form basically a cancer. Again, to identify adult stem cells, we do a few things: attach special markers to the cells and see what kind of cells they generate, see what it becomes. Removing the cells from a living animal, such as mouse, labeling them with special markers, and injecting them into another animal to see what the tissue types they form. Again, we use genetic engineering methods to kind of see what DNA is transcribed. Again, a lot of this is figuring out how to use stem cells. We really haven't done it yet. So there's a possibility of using stem cells to kind of regenerate the heart after a heart attack, to cure diabetes. There's a whole bunch of different things that we do, and there's lots of unanswered questions that we have, even though we've kind of spent billion dollars over. And so some of the things that we see is like, how many different kinds of adult stem cells are there? Ideally, for every single tissue that you're going to have, which is over 400 of them, you're going to have stem cells that are responsible for that. And again, are there a master adult stem cells, one that like embryonic stem cells can make kind of any part of the body? So these are all questions that biologists kind of want to know about. Again, you need to know, so understanding cells and tissues. Cell is the smallest unit of living life that we can have. Number of cells to come together to perform a certain issue are what we call tissues. And so again, we will look at the cell structure and function. You need to know what a cell is, the nucleus, the embryos, the plasma membrane. Everything on the outside of the plasma membrane isn't alive. Everything on the inside is alive. You need to know what a nucleus is and how the cells produce protein, so that's really good. The cells, for the most part, are pretty small. They're going to be less than 100 microns in size. Here's an example of some of the different neural cells that we have in our body, which are relatively large. There's a bunch of different types of cells that we can look at. Again, covering entrance and exits, how things move in and out of cells. And so again, endocytosis and exocytosis, these are all things that you should be kind of aware of. It's how our cells feed. Again, our cells communicate with one another, and they can use diffusible signals like hormones sending back and forth. They use electrical signals. They can use tactile signals. So cells can actually touch one another and perform that activity. And what a cell is surfaces it on can have it. And there's also mechanical signals as well. So a cell under stress will do certain things. A number of cells come together to build what are called organs. Again, the four main tissue types that we see are epithelial tissue, connective tissue, muscle tissue, and nerve system. These are all cells that we would typically see in animals that we look at. You need to know what cells are there. These tissue cells come together to form an organ, like a stomach, like an eyeball. These organs come together to form organ systems, which you would need to know. And so we have things like the excretory, immune system, muscular system, nervous system, reproductive system. These are all things that you kind of need to know pretty much. Again, when we compare stem cells to other cells, they are not as differentiated. And so here's a stem cell. When that stem cell divides, it can do a couple things. It can divide into two other stem cells, which are each capable of self-renewal, or this cell, one of these cells, can become specialized. If both of these cells become specialized, they're not going to divide into anything else. And so that's really critical that you get that, and that's one of the things that we looked at in the first one. Again, stem cell history that we have. We human beings have been using stem cells for a long time, especially in plants. In animals, it's been a little bit more difficult. Again, there are certain organisms that can regrow limbs, legs, and kind of stuff like that. But it's quite a bit harder for animals to kind of do it. Again, we human beings can regenerate. We regenerate our skin. We regenerate our blood. We can regenerate our livers. So in order to understand this, we need to look a little bit about genetics, look at Mendel's. We need to know what a gene is. We need to know what a chromosome is. Again, you need to understand what the DNA is, and so that DNA is a double helix, and we have the sequence across that DNA that covers it. Again, our genes are located on our chromosomes, and so that we physically can map them to where they're at. And human beings are all going to have genes at the same particular loci. Growth factors are things that help cells grow, and we use those growth factors all the time when we are growing our cells. That's why we grow them in human bovine stuff. Again, we've been trying to transplant organs and tissues since the 1950s, sometimes with success, sometimes with not. Why don't we just try and grow the organs that are inside there? The other thing that we have to do is our body's immune system. And what we're seeing is we can transplant organs a lot of times, but we have to kind of shut people's immune systems down so that they don't reject the organisms. In vitro fertilization is this process where we fertilize eggs outside of the body, and then we can put them back inside the body. This is typically where we get a lot of our embryos for our embryonic stem cells. Cloning, and again, this process, if we have a cell that can become any part of the body, we have a possibility of a cell that can become anything that you want it to be. And so cloning and stem cells are definitely things. And so in part 2, we're delving into the stem cell science. I'll probably take about 5 minutes to do this. Again, we want to understand where embryonic cells come from, discover the fascinating properties of embryonic stem cells, and we're going to see what this science does today. And so what we look at here is that all of us develop from a fertilized egg cell. This develops into a zygote and a blastocyst, and it's implanted into the womb, and that becomes a fetus and a baby, and eventually we become an adult. And so when we're looking at embryonic stem cells, we're looking at this kind of early stage here, and we're getting our cells from that stage. Again, sexually mature animals, including humans, produce both egg cells and normal cells. The blastocyte is basically what we're looking at in order to look for the stem cells. And so this is a blastocyst. This blastocyst is basically become all the different cell types in the body. And so this structure right here is called a gastrula, which is basically has these cells in the middle, and it's these cells that can differentiate into these cell types. And in fact, these cells are maybe terminally differentiated, so actually we're not really going to use those cells. Fertilization is critical in doing so. Basically, where we're getting cells is from this blastocyst, and then we can freeze those cells. We can actually take these blastocysts and freeze those for quite a while, and then use those individuals a little bit later. Stem cell properties, again, the properties that stem cells have is that they can divide and grow more or less indefinitely. They can develop into any cell type that's found in the body, that have incredibly lengthy shelf lives, so they can be stored for very long periods without losing their potency. This is, again, freezing them. You can't just put them up on the table. We have a number of different types of stem cells, what we call pluripotent, that become any part of the body. Multipotent can be many things. Totipotent can become kind of anything. And so it's these cells that we are kind of looking at. Again, we can make cells and tissues. We can manipulate those cells. And again, this is talking a little bit about how we do it. This is looking at a process that we call the FACS sorter, and we'll talk about that a little bit in class. This is looking at the research, understanding the possibilities and limitations of embryonic stem cells. Okay, we're not going to cure every single disease right now, right yet with that. The idea was, ideally in the beginning, was that you can just take stem cells and inject them back into the body and they can become any part of the body, but we now know that is not the case. Again, we want to next look at adult stem cells. We can discover where those adult stem cells come from, what can these cells do and not do, and how do stem cells operate in various tissues is the topic of this part of this second. So we can look at what those stem cells, again, your skin and your blood are produced by stem cells. We've been studying those for the most amount of time, especially our blood. And then what we see here is that we have a hematopoietic stem cell, and this stem cell can actually make another oneself, but then it makes a specialized cell that's eventually going to become the red blood cell. Eventually these red blood cells aren't necessarily from here directly, but they are a part of it. And then what we can see is here, this is looking a little bit at how they work. So all these different cell types can actually come from these original totipotent cells, and that these cells become become more restrictive on what they can produce as they get a little bit older. Again, this is looking at a hair follicle. When we look at our skin, it's one of the few substances that are still alive, and we'll take a look at that. Can we make stem cells from these? Again, finding stem cells, this pluripotent, this inner cell mass in the blastocyst is typically where you would find the embryonic stem cells. The adult stem cells are somewhere else. We don't necessarily know where they're at. Again, these blood-forming cells, we know they work because we can take mice and we can destroy the mouse's ability to make its own blood. And when we transport it with these cells can make any of the blood from the mice and from the humans. The brain we don't know as much about. We suspect there's some stem cells in there, but again, we just do not know as much in there. Again, the fetal umbilical cord stem blood cells, I have my kids do that all the time. And again, looking at stem cell sources, we would look, again, we can use human beings. There's a bunch of different things that we can use for these guys. Gotta get going. Other stem cell resources. This is too much. And nuclear transfers, basically what they did to do Dolly. Here's that kind of inner cell mass, what we're looking for, and that cell mass can become anything. You know, again, sometimes we want to bring it to a halt, and we want to put stuff in there, and then, you know, again, those are going to give us organisms. And so, do we want those organisms if we can't really go on to them? And so, again, we can engineer the cells, and that's basically about it. Next semester we will be covering a little bit more. So with that, I will leave you and say thank you.